This week it's Grégoire Herzog from the Université de Lorraine in France. So Greg is probably one of the main reasons I'm doing liquid electrochemistry. I met him when I was only in a third year undergrad in a summer internship, and he convinced me to do science forever. So it's all Greg's fault that we're all here today. So yeah, I've known Greg nearly 16 years now, and we really and truly managed to maintain a relationship of collaboration all the way through. So it's been great. So Greg was in Ireland until around 2011, was it? Something like that. At the Tyndall National Institute, and then he went back to France at Nancy. So yeah, really looking forward to your talk today, Greg, and off you go.
Okay. So thank you. So today I'm not going to talk about liquid electrochemistry. I'm going to talk to you about some of the work that has been going on in Nancy, Université de Lorraine for quite some time. And then when I joined Nancy in 2011, I continued working with the people already there. So there's the field was initiated by Alain Walcarius, and I'm also working with my coworker Neus Villa. And some of the work I'm going to present today was done by PhD students who were working with me. So it was Tokir, Deomila, and Himanshu. So I'll start with why are we trying to make mesoporous silica thin films for electrochemical sensors? Okay, so the interest for silica is soil gel chemistry has been around for many years and it's been known since the 19th century. And it's a very rich chemistry with many organosilanes that are available, so you can functionalize and give functions, chemical functions to your to the silica that you are synthesizing. You can get some rigid porous structure, so it gives a good base for modifying an electrode, and it can host reactants and maintain their activity. And there is plenty of papers where proteins have been immobilized in silica matrices and shown that they kept their ability, their catalytic activity, enzymatic activity. The main problem is that silica is an insulator, and when we want to do electrochemistry, it's quite a problem. So what we need to do is we need to be able to deposit thin films, porous films that are stable on conductive substrates. So then we can do electrochemistry with them. So the soil gel chemistry is basically a two-step process. So how to get silica is the first step is hydrolysis. So we start with from tetrahydroxysilane or alkoxysilane, and we're going to hydrolyze the SiO bond to give us a silanols group. And then this, when you have two silanols group, then they can react and form a SiO-Si bond. And so this is the condensation step. And these two steps are optimal at two different pH. So at pH 3, we would like hydrolysis will be favored. And then at pH 9, between pH 9 and 10, we will favor the condensation. Also, it can happen at very acidic, in very acidic conditions below pH 0. So, and we're going to prepare those mesoporous silica films by playing with these two optimal pH there. So I'll show you a bit later. So, more mesoporous silica by electrochemistry, there was two key advances in the 1990s. The first one is a Nature paper from a researcher in the Mobile Oil Corporation. And what they did is they mixed silica precursors, surfactant, cetyl-trimethylammonium bromide. In this case, then they found the right conditions to form micelles and condensate the silica around those micelles, and they form the MC141, the well-known mesoporous material. And here you have a SEM image of the of these mesoporous particles that were formed in this way. So this was a new way to make mesoporous silica using a surfactant. And then also a second study that was important for this work was the electrodeposition of salt gel. So there was work that was done by the group of Daniel Mandler in Israel, and he showed that if we were applying a reductive potential at an electrode surface, that in the presence of hydrolyzed silanes, then you would electrodeposit silica, and the amount of charge that was passing through the electrode was determining the thickness of the film. So taking these two studies together, new methods to form mesoporous silica thin films was developed in the group. So what we were doing is we took hydrolyzed silanes that we mixed with cetyl trimethylammonium bromide, so the surfactant, and with some electrolyte. We immersed the electrode and we applied a reductive potential that was reducing protons and reducing water. And so we were changing the pH in the vicinity of the electrodes, and then we were getting a pH gradient. So close to the electrode, we had basic conditions, and in the bulk we remain at pH 3, which was the hydrolysis pH there. And as a consequence, the thin film was growing from the electrode surface towards the bulk. And but because we were doing this in the presence of surfactant, the negative potential that was applied at the electrode surface forced the cationic surfactant to form hemispherical micelles on the electrode surface, and the silica was condensing around those micelles. And as the silica film was growing, the micelle was growing along with it up to a certain limit, about 100 nanometer, 120 nanometer of thickness there. And then beyond that, we started forming some silica beads. From some silica beads. So here in this case, the electrochemistry offers both the control of the pH in the vicinity of the electrode, which is important for the growth of the silica, and then it also allows to organize the surfactant at the electrode surface, and then so we were getting these the mesostructure. So this is an image of what the film looks like. So if we look if we look here, at the cross section of the film, you see that you have the glass slide. Then here you have the ITO, the ITO electrode, and then on top of it you see the silica film. And on top of the silica film, if you deposit too long, then you see that the silica beads are being formed there. So we are trying to avoid the formation of silica beads there. And the surfactant, and we can play with the diameter of the chain length of the surfactant, and we can have pore diameters between 2 and 3 nanometers, and for thicknesses that are ranging from 50 to 100 is maybe a bit too large, but 150 for sure, yeah, without silica beads. And then after you have TM image, top view T image of the film. So each white dot here is a pore, and you can see that is in hexagonal compact arrangement there. And here you have a section of the film where you have the electrode here, and you can see that the pores are going all through the silica film thickness. So what is very important here is by doing this electrochemical synthetic method, you have pores that are perpendicular to the electrode surface, so the electrode surface remains available for the analytes or probes to diffuse through the film and reach the electrode to be oxidized or reduced there. Here. Another characterization technique is GIXD, where we could verify that we had a long distance order of the film, so we maintain these arrangement over a large surface area. And here we can see that if we were if we were seeing rings there, it would mean that we had many silica beads on the top of the surface. But here, if we have the correct conditions and we manage to control the film formation without the appearance of these rings there, and so it shows that we have that the deposit is only the film and not beads on top. So we can electrogenerate these on many substrates, many conductive and semiconducting substrates. It works very well on ITO, indium tin oxide, or FTO, fluoride tin oxide, because of the presence of oxygen groups on the surface that can form covalent bonds to the with the silica film. But sometimes we need to help the adhesion with using a molecular glue. So here in this case, we were first electrographing aminopropyl tritoxysilane, so to form a covalent bond between the nitrogen atom and the carbon electrode. And then we had selenol groups that were available to form bonds with the growth of the silica. And so here's the SEM image of a film that used the APTES as a molecular glue, and you can see that we have a smooth and homogeneous film. And the only reason we realize there is a film is because we have some silica beads on top there. If we don't use this APTES layer to immobilize the film, you can see here that the film is not properly adhering, and here you see the bare glassy carbon with broken bits and pieces of the film there. So it's really important to be able to have this APTES layer to form the silica, homogeneous silica film. If you change the substrate, for example, if you want to do on the gold, you change the silane, and instead of using an aminosilane, you can use a mercaptosilane to form a gold sulfur bond on the surface of the electrode, and then you have a much better stability of your film. So, and one of the good things of the electrochemistry is it allows us to control the thickness of the film because it's linked to the amount of charge that is passing through the electrode. And so if you have a rough surface, so here you have a rough surface where you have hills and valleys, and you can see that with electrogeneration you can follow the roughness of the film. But if you do this in spin coating or another evaporation induced method for the sol gel process, then you will completely fill the gaps there, and then you would lose the structure of your electrode. So this is one of the good things, also good reason to do to use electrochemistry to deposit those silica films. So now I'm going to talk a bit about the electrochemical characterization of those films. So so here is we use the ruthenium hexamine as a redox probe, and here is the signal at the bare electrode. And so when we deposit the film, before we extract the surfactant, this is a schematic on how the surface looks. So we have the surfactant is still present inside the pores, and the electrochemical response for the probe is completely suppressed by the presence of almost completely suppressed by the presence of the silica and the surfactant and the surfactant inside the pores. Then if we do the extraction of the surfactant, which can be either by calcination or by acidic ethanol solution, we can remove the surfactant, and then you get the CV is almost back to normal, and so it means that the electrode surface is available again for the reaction, but the film is still there. So we're only removing the surfactant there. And so the first things that when we tried to do when we wanted to use those for sensors is to look at the charge selectivity, because at pH 6 the silica is negatively charged. So we looked at a cationic probe, so paraquat, a neutral probe, the catechol, and an ionic probe, the diclofenac. And so here on all these cyclic thermograms, the blue line is the bare electrode, the red is the modified electrode, and the black is the blocked electrode before surfactant removal. And you can see with paraquat that we get a larger signal with the film than with the bare electrode, suggesting that we have some electrostatic interaction between the negatively charged silica layer and the cationic probe. If we look at the catechol is neutral and is not much bothered by the presence of the film. We can see that the peak are slightly broader and a bit lower, but we still see a good signal. Whereas in the case of the diclofenac, the electrochemistry of diclofenac is completely suppressed by the presence of the film. So this suggests indeed that we have electrostatic interaction and electrostatic accumulation of the probe in the films. We also the effect of the pH on the signal of paraquat, and if we go at a pH around 1, you can see that the film is going to be positively charged, and here we completely suppress the response for the paraquat due to electrostatic interaction. The isoelectric point of silica is between 2 and 3, so it's like it will be indeed positively charged. pH 6 is what I showed you previously. Is the probe is cationic and the walls are negatively charged. And then if we increase the pH above 8, and here is an example with 10, we still get a signal here at the modified electrode, but it's shifted to more negative potentials. So it suggests that it's more difficult to have a reaction, redox reaction occurring, and the signal is lowered compared to the pH 6. And what happens is that we're starting having some dissolution of the silica walls, and then we're losing part of our signal there. So what we have, if we want to have an optimal signal, we should be between pH 6 and 7 to be sure that we have a negatively charged walls, and that we don't start dissolving slowly the silica films. And so the idea was just to how can we use this silica to improve the sensitivity and the selectivity to make electrochemical sensors. So we looked at the effect of cycling paraquat. So we did a series of 50 cycles at a modified electrode and a bare electrode, and you can see that at the modified electrode you see the signal is increasing before it reaches a plateau. So meaning that we are with time and with the number of cycles we are accumulating the probes inside the film. And then if we did this for a series of concentration ranging for 10 micromolar up to 8 millimolars of paraquat dissolved in the solution, and we could see that at the low concentrations we were always having a greater signal for the modified electrode and for the bare electrode, suggesting that we were pre-concentrating the paraquat inside the film, and then reaching the higher concentration between 2 to 8 millimolar, we were getting close to the signal of a bare electrode, suggesting that the amount of free paraquat that was diffusing freely through the films was more significant, or significantly greater than the amount of silica that was absorbed on the silica. The amount of paraquat that was absorbed on the silica was. And then so we could verify this by doing some cyclic voltammetric studies and scan rate studies, and you can see that at the low concentration of 20 micromolar, we do have absorption behavior with a peak-to-peak separation between 10 to 20 millivolts, and the peak current dependence was linearly dependent on the scan rate. When we were increasing the paraquat to 1 millimolar, we started to see a diffusive behavior with a larger peak-to-peak separation, and the scan rate that was dependent on the peak current was dependent on linearly dependent on the square root of the scan rate. So when this, we tried this accumulation for different types of probes. So we tried, I showed you paraquat here in red. We tried diquat, which is here in green, methylene blue, and then ruthenium hexamine, and then ruthenium trisbipyridine. And this is, the solid lines are the signal after 50 cycles at the modified electrode, and then the dashed signal is the signal after 50 cycles at the bare electrode. And you can see that for all probes, we do have an increase of the signal due to the presence of the silica film. But it's not to the same extent. So for instance, here we calculated the managed fire. You can see that for paraquat and methylene blue, we have we multiply by 10 the amount of the peak current, whereas with diquat and ruthenium tris-BP was about 4 times, the signal was about 4 times greater, and then from ruthenium hexamine, it was still a bit of absorption. The signal was improved by a factor 2. So this shows that it's not only this accumulation is not only due to some electrostatic interaction between the negatively charged walls and the cationic probe. There is also some other parameters that play a role. Just to show also that we had some like the absorption. Here we plotted the peak current as a function of the scan rate in logarithmic scale, and so a slope of 1 would correspond to that absorption behavior, and a slope of 0.55 would correspond to a diffusion profile. And you could see that for the paraquat and the diquat, we reach a slope of 1, and then for the the ruthenium hexamine and the ruthenium tris-bipyridine, we were in a mixed signal there for 20 micromolar, and then when we increased to 1 millimolar, then as I showed you before, for paraquat, everything is a mixed diffusion absorption behavior. So one of the reasons why we are absorbing more paraquat, diquat, or methylene blue compared to ruthenium hexamine, for instance, could be due to the hydrophobicity or the organic nature of the paraquat. For instance, is that we need to remember that we are in a confined space where about 2 to 3 2 to 3 nanometer in diameter and 100 nanometer approximately in height. So those organic molecules that are going through the pores that come with a hydration shell, and one of the reasons why they adsorb very strongly and very well to the silica walls could be due to the fact that they have less water molecule would be needed to hydrate them if they are adsorbed, and so you could get more. This adsorption could be thermodynamically favored, and also what you could have is you could also have some pi-pi interaction between these hydrophobic or these organic molecules, and that would make probably create some islands of organic molecules in the wall on the silica walls, and this could help explain the formation of those like the greater accumulation of these organic probes in the films. We also looked at the effect of the counterion. So when these organic cations these cations are going inside the pores, they are accompanied with their with the counterions. So if we use sodium Bf4- as an electrolyte, Bf4- is a rather large anion, and we absolutely have no signal. We cannot reduce the paraquat. And then if we change the ions, then we can see that depending on the ion, we have quite a large distribution of peak current. The best is recorded for nitrate as a counterion, and you can see that this is the trend that we have with the current for nitrate is the largest, and the current for Bf4- is the lowest, is to 0, and then. But there is no direct correlation with the Hofmeister series, the ionic radius, and the conductivity. So I'm sure all these play a role in a way, but one of the reasons also could be also due to the geometry of the ions, of the anions. And paraquat, for instance, this is with paraquat, and paraquat is a rather flat molecule, and so is nitrate. And so it's probably that it's for geometric reason it could be intercalating with between paraquat molecule, and we could pack more paraquat in the thickness of the film there. So all these are just like hand waving explanation. I don't have any scientific and any experimental proof to this, but this is a gut feeling. So like we would need to be able to confirm this. We would need able to do some perform some molecular dynamics to investigate the hydration and adsorption of paraquat and the other probes in these nano-confined spaces, and the effect of the interaction of the electrolyte ions with the probes, and also maybe like looking at locally on the adsorbed species, and maybe look at which chemical groups of the adsorbed species is being affected by the silica film could also might give us some information there, looking at some intermolecular interactions. So these are things that we had planned to do, and but we were still running behind because of the lockdown, and we haven't got back to full, let's say, experimental capacity yet. Okay, one of the other things that we have been looking at is the effect of the ionic strength on the electrochemical response of paraquat. And so here is the square wave voltammetry of paraquat for 3 different ionic strength of 20 millimolar, 100 millimolar, and 70 millimolar. And we found out that the best ionic strength was 70 millimolar, and then for here is you have the peak current as a function of ionic strength, and you can see that we have the maximum reductive current for 70 millimolar. And the fact that this is dropping at lower ionic strength is not due to a loss of conductivity in the solution, is because it does not affect the bare electrode as well, and it's more to do with the what we think is due to the Debye length in the system, because in the ionic strength we're looking at and the pore diameter we're looking at is like the Debye length and the pore are in the same order of magnitude. When you are at ionic strength that is below 70 millimolar, your Debye length is greater than the radius of the pore. So you're probably excluding anions from the pore, and so it is not kinetically favored. The reduction of the reduction of paraquat is not kinetically favored, and then so we see a loss in the peak current in the signal. And then if your ionic strength is higher than your pore radius, then your Debye length is smaller than the pore radius, so it's fine. The anions can enter and circulate in the free volume, but the reason why the signals start decreasing is probably because the paraquat enters in competition with a large excess of sodium ions and electrolyte ions, so you're probably losing signal this way. So we found that the optimum was around 70 millimolar, which coincide more or less to the Debye length and the pore radius coincide. And then when you using all these optimized parameters, you manage to have a sensitivity that is improved about 24 times. So compared to the bare electrode, and then so you can start looking at real applications. So what we did is we did some square wave voltammetry of a water sample from the Meuse River. So we spiked it in the we spiked it with paraquat. We did not find any paraquat in the Meuse River, but Meuse is a rural area with lots of fields, so it was quite high ionic strength. It was we estimated to be about 4 millimolar, and it was quite a large concentration of nitrate there, which was serving our purpose here in helping detecting the paraquat. And you can see that at the blue electrode we could not see any signal, but at the modified electrode we could see a signal from 10 nanomolar to 100 nanomolar, and we calculated the statistically limit of detection around 12 nanomolar, which is below the permissible maximum level of paraquat, which is 40 nanometer. And this was done without any addition of background electrolyte, just using the pure the water as it was collected. So this is an example of what could be reached in terms of sensitivity with this Mesoporous silica electrodes. So I'm going to talk briefly, I guess I still have some time, about how we can introduce some selectivity to our systems by looking at the electrochemistry of cysteine. You can oxidize cysteine is an amino acid that has a thiol group here, so it's the chemical structure, and you can oxidize it. And if you look in the literature, the standard potential of cysteine is about minus 0.25 volts. But the thing is, if you try to do this at an electrode, you have an over potential that is greater than 1 volt. And here you see the oxidation of cysteine is about 1.1 volt or so, and it's not a very well defined signal. So what people are doing is generally they introduce a mediator to oxidize the cysteine, and so this way you can lower the overpotential. So here an example is a ferrocene. So ferrocene is oxidized at the electrode surface to give ferrocenium, and ferrocenium is being reduced by the cysteine, and then it's regenerating ferrocene that can be detected again at the electrode surface. And here you can see the cyclic voltammogram of the cysteine at the in the absence. The cyclic voltammogram of ferrocene in the absence of cysteine at a bare electrode, and here after the addition of nearly 1 millimolar of cysteine, and you can see that you have an increase of the signal of the forward peak, which correspond to the regeneration of ferrocenium in ferrocene by the cysteine, and then you can see that the reverse peak is diminishing because there is no very little reduction of ferrocenium as it has been consumed by the cysteine. And so you have a different type of thiols. And then if you're in a large excess of thiol over a cysteine, you can determine some kinetic parameters. And so we did this for homocysteine, which is the same as cysteine, but with an extra CH2 group in the side chain of the amino acid. Cysteine, glutathione, which is a tripeptide which has a cysteine group, and then methionine is a sulfur containing molecule, but it's not a thiol, and we could see, we used it as a control, and we saw that we have no reaction. So just to be sure that it was the cysteine, that was the thiol group that was reacting with the ferrocene. And so when we did these experiments, similar experiments at the electrode that were modified with the silica. Sorry, there is a mistake here. The bare is in black and the modified is in red. Sorry. So and you could see that the signal was lowered lower at the modified electrode than at the bare, showing that for cysteine the presence of the silica film was having an effect on these on the kinetics of the reaction, whereas in the case of the glutathione we had similar limiting current, suggesting that maybe the glutathione was not so bothered by the presence of the film, and so suggesting that maybe glutathione was not entering the pores to react with the ferrocene, but it was rather the ferrocenium that was diffusing outside to the bulk of the solution. And anyway, the kinetics was sufficiently slow for the glutathione not to be bothered by this extra diffusion, let's say, of the ferrocenium through the film. And so we thought maybe if there might be something there that we could induce some selectivity there, but to do this we would need to stop the ferrocene to diffuse freely in the solution. So, but to attach it covalently to the electrode to the electrode surface. So what we did click chemistry using a protocol that was developed in the lab as well, where well, not the protocol for the click chemistry, but the click chemistry on the Mesoporous silica. And so we were having an azide silane that was synthesized. Then we were reacting it with an alkyne that was functionalized with a ferrocene to give this kind of structure there. And we could have the ferrocene that was attached there covalently to the electrode. So it was not diffusing freely anymore in the solution. And so only species that were at the mouth of the pore that was diffusing inside the pore could react. So then we did some cytometry in the absence of thiols. So this is the line in black here. And then when we added the cysteine, we could see that the signal was slowly increasing on the oxidation side, and then it was slowly decreasing on the reduction side as well. So suggesting that indeed cysteine was being detected at those ferrocene functionalized electrodes. But when we did try with glutathione, we see no significant differences with the CVs with the addition of glutathione. So then after we placed the electrode in a flow cell, and we did some injection of cysteine ranging from 3 micromolar to 100 micromolar, and we could see that we'd had an increase in the current recorded. And then when we were doing addition of glutathione and cysteine, we could see only that we had, like, for 50 micromolar 50 micromolar of glutathione, we had a peak that was barely going out of the noise, whereas with cysteine we had a much more stronger signal. So this showed that we could induce some selectivity to the system based on the size of the pore and on the reaction that was occurring at inside the film. So this is all what I wanted to present for today. So I've shown you how we can make and characterize those silica films, and how we can use them to improve the sensitivity, and how they how we can play with their nature to improve the sensitivity. We can use them for real sample applications. Real sample application, and then we also can introduce some selectivity using some click chemistry there. Okay, so thank you for your attention.
Thanks, Greg. Brilliant. Like, your summary slide there is ideal. Like, with your modified electrode, you can do exactly what you want for your sensor application. So the sensitivity and the selectivity. So yeah. I'll open it up now to the floor if anybody has any questions.